So I think we can start slowly. I press the record button. Merve, you can introduce.
Okay. Hi, everyone. Thank you for all being here and welcome to today's online FoRCE seminar. So let me give you some information about FoRCE little bit. FoRCE is dedicated to provide free, high-quality outreach events and online seminars to reach broader robotics and control engineering community around the world. Thus, we periodically invite distinguished people like Dr. Del Vecchio to give their seminars on recent research results related to robotics and control engineering. The aim of the FoRCE is connecting academicians and government industry researchers with each other through the research and education discussions. So we cordially hope that you will enjoy all the FoRCE events and you will find them highly valuable for your own research. By taking this opportunity, I also would like to talk about the support from IEEE Control System Society, where you can also find the past FoRCE talks and many other talks at the IEEE CSS video library webpage. A little bit information about the WebEx. You are all muted during the presentation, and if not, please mute yourself. If you want to ask questions after the presentation, you can do this by simply unmute yourself, or you can just type your questions to the chat box. And this session is being recorded and to be posted to the FoRCE as well as the IEEE video library webpage. So if you don't want your voice to include it to the video, just type your questions to the chat box. So jointly with Tansel, Yucelen, we are very proud today to host Dr. Domitilla Del Vecchio. And Domitilla Del Vecchio received a PhD degree in Control and Dynamical Systems from the California Institute of Technology, Pasadena, and the Laureate degree in Electrical Engineering from the University of Rome at Tor Vergata from 2006 to 2010. She was an assistant professor in the Department of Electrical Engineering and Computer Science and in the Center for Computational Medicine and Bioinformatics at the University of Michigan, Ann Arbor. In 2010, she joined the Department of Mechanical Engineering at the Massachusetts Institute of Technology, where she is currently professor and member of the Synthetic Biology Center. She is an IEEE Fellow and a recipient of the Newton Award for the Transformative Ideas during the COVID-19 pandemic 2020. The 2016 Bose Research Award, the Donald P. Eggman Award from the American Automatic Control Council 2010, and the NSF Career Award 2007, the American Control Conference Best Student Paper Award 2004, and the Bank of Italy Fellowship 2000. Her research focuses on developing techniques to make synthetic genetic circuits robust to contacts, and on applying this biosensing and the cell fate control for regenerative medicine applications. Well, for all of you here, I would like to thank Domitilla for participating in our forum as a speaker. Domitilla, as you have the presentation board, please go ahead and start whenever you are ready.
Thank you very much, Merv, and thank you for the introduction and for the invitation to speak here. I assume you can hear me well?
Yes.
Okay, sounds good. So today I will present an overview about some of the control theory flavor and tools that we have been working on for the past, I would say, 10 years for challenging problems that we find in engineering biology. And in particular, here I have a short outline. And this is really meant to give you a sense of the journey that it takes to get to the point where we hopefully one day will be able to engineer biology just like the way we engineer a modern computer. So I will have a short introduction, engineering biology, why and how, since I assume many of the people here are not familiar with the field. Then I will point out some of the critical challenges when we talk about modular composition. And then I will hit on two technical problems that we have been focusing on in the past 5 to 6 years, which is the problem of load rejection, which is very similar to what's found in electrical and mechanical systems, but the physics of biology is so different that we had to come up with slightly different ideas that we typically see in electronics. So let's start with the general introduction. So to give you a brief historical overview, engineering biology, also called synthetic biology, is based on genetic engineering, which is really the field by which you can manipulate DNA in a number of different ways. And there was about a decade, starting in 2000, where people were focusing mostly on engineering simple circuits just to understand biology. There was an important transition in the community around 2010, where people started to realize that the technology was mature enough to think about engineering systems to do something useful, so useful decision making. And when I talk about this, I always like to show this movie. This is a neutrophil, is a white blood cell that is chasing a bacterium in the bloodstream. And when I saw this movie from the first time, I was very excited because I thought this is a great example of a natural sensing, computation, and actuation all in a feedback loop, right? That is working perfectly. So people have been thinking, what if we can extract and re-engineer these sensing, computation, and actuation components in order to tackle a bunch of concrete problems? So here is just a small set of examples. So these go from essentially curing disease. So there are researchers that are focusing on engineering bacteria that have the capability of sensing the location of a tumor once they are in a bloodstream. So they are engineering them so that once they sense the tumor, they lyse open and they release killing agents. So that's a way to targeted killing of cancer cells. There is a whole area which is developing right now about designing genetic circuit to control cell fate, which is going to be critical for fields such as regenerative medicine. And then there is, as we speak, really already startup companies that are coming up about engineered microbiome. So the idea is that you take the bacteria from your guts, you re-engineer them, for example, to sense stress markers, and they detect when some potential drug release is necessary, and then you put them back and they help you, for example, deal with stress or difficult workloads situations. And then there is a whole area about biosafety where people are thinking of engineering cells that they can detect threats such as viruses, poisons, and counteracting them. So that said, how do we program cells with new functions? So here is a really high-level introduction, which is showing you how what we call circuits become encoded into DNA. So the idea is that you have your bacterium. The bacterium has circular pieces of DNA called plasmids. You extract them, you cut them, and you insert synthetic DNA, and then you put them back in the bacterium. So the synthetic DNA encodes genes that express proteins that regulate the expression of other genes. And this way you can create circuits by inserting synthetic DNA that has multiple genes regulating one another. So in this process, the key functional module is the genetic module. So it's a dynamical system that has essentially the process of transcription by which a gene becomes messenger RNA. And from messenger RNA, through translation, it becomes a protein. And this is a system with inputs. So it can get activators, meaning proteins that help gene to be expressed, or it can take repressors, meaning inputs that prevent expression of the gene. So what people have been doing, have been using this genetic model as starting building blocks of genetic circuits to create networks. What I'm showing you here as an example is a 3-node networks with 3 repressors in a ring function. This was the one of the first 2 networks built in 2000. And what I'm going to show you here, it's a movie that represents, is showing you cells, and the fluorescence of cells is essentially proportional to the concentrations of one of these proteins. So if you track a single cell, you can actually see that this system is an oscillator, okay? So this was published in 2000, and since then, there have been a number of additional circuits built and characterized, and at this point, from this talk, we will characterize and describe the systems by differential equations, okay? So systems that takes inputs and give outputs, they are described by ODE, and the key assumption for the entire talk is that molecular counts are sufficiently high so that a differential equation description that describes the mean levels of these molecules in the cell, it's meaningful. So let's look at one of the critical challenges that people have been facing, which is modular composition. So when we engineer normal systems such as electrical, electronics, computer, and even computer networks, describing a sophisticated system as the composition of simpler modules with specified inputs and outputs relationship, it's a great helper to scale up the complexity of design, because we can forget essentially what's inside the input-output map and just focus on the connection rules in order to predict the behavior of the whole system. And in this process, feedback has been critical to maintain input-output properties of modules, provide simplified abstractions, right, for layer design. So based on this picture, people have been tempted to follow a similar notion also when engineering biology, going from pieces of DNA to core modules, such as I've shown you before, and then pathways combining together, and then putting into cells, and then cells coming together to form tissues. And this has been very tempting. And the hope is also that if you look at how quickly we can synthesize and sequence DNA, it's at comparable speed at which the number of transistors per chip has been increasing. But one key problem that we have is, although increased scale is becoming possible in engineering biology, modularity, being critical to manage complexity and time, is often failing, okay? The unfortunate scenario is that when you combine these genetic modules, let's take an example, the input-output properties of this module are affected by surrounding systems. So what the designer typically do is to redesign everything that was in your system after a new piece is added. And that's why for a circuit with just 11 genes, it takes one PhD thesis for about 5 to 6 years to get it done. And it only works in very controlled laboratory conditions. So what are some of the reasons why modularity is a challenge? There are many. Here I will focus on a few of them that we can represent nicely within systems and signals description. So one big one is the loads, okay? So anytime you connect an upstream system with a downstream module, there is a loading that comes from downstream system. We will see the physical origin of those for biological systems. And so this essentially is changing the behavior of the upstream system once it gets connected. That's one way modularity fails. Another way that was discovered more recently by which modularity fails is the fact that all these modules are placed in the cell to function, and they are using cellular resources, okay, such as ribosomes, RNA polymerase, or any other enzymes. And people have been assuming that these resources, there is plenty in the cell, but the reality is that that's not the case, especially as you keep increasing the size of the circuits. And so modules apply load to the cellular resources, and this can create actually very subtle couplings between modules, which are very difficult to untangle. Then you can take it further. Modules have off-target effects with the cellular circuitry. The cellular circuitry can interact with the modules, so there are all these perturbations that come up that affect the module behavior. And beyond the physical origins of this, if you view the problem in this way, you can view this as perturbations that are acting on your module, and then you can ask the question, how do you make your module robust, right, to this perturbation? It's definitely a control system design problem. And that's why, essentially, for these and other issues, there has been a tremendously increasing trend of people in the control community that are starting to be interested, right, in problems of design in synthetic biology, because in many of these problems, actually most of them, robustness is the issue, and control design has been critical in engineering robustness in most of our common engineering systems. So the first story I'm going to tell you is about how we handle the issue of loading in between essentially genetic modules. So how is it occurring? So here is an example. So this is a genetic oscillator, and it's having this spiky oscillations. So what you can do with an oscillator, you can connect it to a downstream system, for example, to transmit or synchronize downstream processes. But when you do so, you'll find out that the oscillator behavior is destroyed. And this makes sense because physically speaking, the way the interconnection is made is by this protein A traveling into the downstream system and therefore being sequestered away from the upstream system, and therefore the interactions in the upstream system become weaker. So loads change the behavior of the upstream system, and this way we fail to transmit the signal. So a general systems and signal representation of these loads in general in biology has been through this retroactivity formulation. So here we are representing a system with intended inputs and outputs, but then we had two arrows here, S and R, that denote essentially the load that downstream systems are applying to your system in question, and R is the retroactivity to the input, which is how the system is changing, okay, the behavior of the upstream system once it's receiving the information in you. So if you take this formulation as your lens by which you revisit, right, the problems when you interconnect systems in synthetic circuits, then here is the picture. So assume that you have a general network here, and again, each node, as we said in the network, is our module, which is a system that takes a regulator inputs and give proteins as output. So you can ask a very simple question: how is this behavior going to be affected once you connect it to the other system? And what you will see is that once you connect your system to a downstream system, what's appearing on the dynamics is an additional reaction flux, which is really your retroactivity to the output S, which appears as a time-varying disturbance on your input and has a rather severe effect. So these are experimental results that show your isolated system behavior and the behavior of the system when it's connected. So by just looking at these figures, you see that the connected system is essentially having a decreased bandwidth, right, compared to the isolated system. And there is a rich theory that studies that. But what I want to get at is, how do you fix this problem? Okay, so this is the insulation device story. So once we, of course, came to this problem, then we said, okay, so let's divide the upstream system from the downstream system and interplace an insulation device, which is a system that essentially has the ability of rejecting the perturbation, meaning the loading that the downstream system is applying. Well, this is a standard disturbance rejection problem, as we have many, right, in control system design. And of course, the first principle that comes to mind to apply is high gain feedback. But in the next slide, I just want to convey the notion that even this is the obvious one to start with, because of the structure of the biomolecular systems that we can actually build and implement, we had to change the strategy, okay? And that's why we came up with a new way to do disturbance attenuation, which is easy to implement in biomolecular networks. So how did the story go? So of course, we started right with this picture, which is what I teach probably in the first lecture of an undergraduate level feedback control course. And What happens here, right? You know that if S is your perturbation, you have your input and your output. If your high gain, G, right, is sufficiently high, the output is going to be insensitive to the disturbance. Good. So we wanted to see if we could implement this in a molecular system to create this insulation device. In order to do this, we revisited this block diagram, and you can rewrite it as the combination of two actions, a large input amplification and a large negative feedback. The reason why it's useful is because if you look at the textbook model of what is called a phosphorylation cycle, that's exactly what it is doing. Okay, so phosphorylation cycle is a molecular system that takes an input U, which is an enzyme, it's called a kinase, which is converting a protein X from inactive to active. And once it's active, then it can go and transmit the signal to downstream systems. So essentially, in the system, you have a large input amplification that comes from this interconversion modulated by the input, and the negative feedback here, which comes from the backward reaction. So if you look at a very simple textbook model of the system and you write the ODE, what you will notice is that, so your input Z, you have your output Y, which is your X star here. You have your disturbance S, which is the state dependence retroactivity. And then where are the gains, G and G prime? They are here. So the gain G and G prime, they are essentially proportional to the total amount of X and P that you have in the system, which practically speaking, they're very easy to tune. So this could provide, in principle, a system where we can tune the gain, G, as we want, and assess the insulation property. So once this was done, and to give you a sense of the timing here, this was about in 2008, 2009. We went to our collaborator, and we were very excited, and he said, look, you know that in reality, the phosphorylation cycles never appear on their own. They always come in complicated networks. And he told us that the simplest, this was Ron Weiss at the time, the simplest network he could build based on this principle was this one. So he gave us this model and asked us, well, can you make this work as an insulation device? Clearly, there is no clear negative feedback system here. So how can we handle that? So we started modeling the system. We had 60 state variables to model that. And to our surprise, we found that it actually had a very good insulation property. Why is that? We figured out that this was because the system have a very fast timescales compared to the timescale of the input, which comes from gene expression. So these signaling systems are very fast. Timescale separation about 100-folds. So here then we had the motivation to come up with a framework for retroactive attenuation that could be easily applicable to design of biomolecular insulation devices. So if you take a general system of your insulation device without disturbance, okay, you have your input U, and then you have your output Y, and you have no perturbation. Once you connect it, now your output Y dynamics is going to be affected by this perturbation coming from the loads from the downstream system. The idea here is that if you have this G1, which is very large, capturing the fact that the insulation device dynamic is much faster than the dynamics of the input, and you have this nice property between M and N that essentially is telling you that the connection is closed. Everything that goes into the downstream system is coming away from the insulation device. So there is nothing that goes out of this connection. Then you can prove that, of course, under stability conditions, then if G1 is sufficiently large, doesn't matter how large is your disturbance, you are going to have that the output Y in the connected system is going to follow what you had in the isolated. So you are attenuating retroactivity. So at this point, we went back to the system that our collaborator gave us. We reanalyzed the 60+ state variable system, and we looked at the timescale separations. and we found that there was exactly the same structure that we had in our model, okay? And the system was stable. And at this point, we could essentially derive right away that the system could work as an insulation device. That's when the system was built, okay? And going back to remind ourselves what the problem was, we had this upstream system connected with the downstream system, having discrepancy here due to the load that the downstream system is applying on your output X. So what we did, we took our insulation device, we called it the load driver, and then we reperformed the experiment where the upstream system was sending the signal with and without a downstream load, okay, to an output module. So here again is what we had before the insulation device, but once you put in the insulation device, you essentially have perfect retroactivity attenuation. So this was essentially the conclusion of the story on insulation devices. So now we know that if you want to transmit something by the feeding loads, you have to ensure to have a stable buffer that has really fast dynamics. So that said, we were at the point where we were pretty satisfied. This was 2014 when a new issue came about, which is this issue of cellular resources, okay? So up to that point, it was about 2014-15, people were pretty convinced that the cell could provide a very large amount of required resources for functioning, so one shouldn't worry too much about dragging these resources down. However, one of my students at the time did this very simple experiment. So I went to the lab and he took a 2 genetic module system. So you have an input is activating A and A is activating B. So it's an activation cascade. And you would trivially expect this to be the input to output steady state relationship. But when he did the experiment, he actually got this. So we saw that, yes, the output was going up, of course. B was going up as your input was increasing, but at some point it was going back down to very low levels, almost barely detectable. So this was rather surprising what was going on in the system. So that's when we went back to the drawing board, try to understand what's happening. So if you put this into the context of the cell, so every node is a genetic module that, as we saw before, it comprises a transcription and translation step. Each one of these steps is taking resources to function, okay? So if you have these resources that are coming into both node A and B, but you're asking your input to drive all these resources to A, then less are left for B, and therefore B can ultimately go down. So then we came up with a framework to try to predict this effects. So the rationale is that if you look at the interactions between these resources with their processes, meaning transcription and translation, they're very fast. So you can assume quasi-steady state interactions there, and the interaction between the modules is the slow one. So if you only focus on the slower dynamics, considering these quasi-steady state assumptions, you can essentially go from a description of your system where each one of your nodes is taking a regulatory input, denoted UI. This is your regulatory function. And these are multiplied by this parameter that are proportional to the amount of resources you have in the cell. If you consider the conservation loss, okay, and therefore limitation of resources, you are actually going to be able to prove that this network is actually equivalent to a network where there are added hidden interactions popping up. So for example, what we have here is that if you have a node that is regulating another node, it's taking away resources from your node in question, and therefore it's going to pop up here in the denominator. Most interestingly, we were able to nail down what is the parameter that regulates the strength of this hidden interaction, which we call the resource demand coefficient, which is easily tunable. So now this becomes the new effective regulatory function. And what you can see is that you can come up with the effective interaction graph. So the effective interaction graph is now composed of a rescaling of the black one, which is the intended regulation graph, and then some hidden interactions pop up. So these hidden interactions have a property that is not dependent on parameters. So anytime you have a node which is activating a downstream node, it becomes an effective repressor for any other node just because it's taking away resources from it. Similarly, if you have a node that becomes a repressor for a target, it's becoming an effective activator for any other node. And then you have that the strength of the intended interactions becomes just weaker, but it never changes sign. So if you take this into account, we went back and looked at our cascade system where we had this expected behavior. But now if you account for hidden interactions, we also see that we have these hidden interactions here with strength measured by this resource demand coefficient that if you look in parameter space given by DNA copy number and the strength of translation, you can tune it very easily. So this is what the model predicts. We did a bunch of experiments, okay, starting from this region in the system, building a bunch of cascades with varying parameters, and we could see that we could essentially ultimately recover the intended behavior of the network by going back to a situation where these resource demand coefficients have low values. So the bottom line of this is that now we are using this resource demand coefficients J to essentially tune and hopefully minimize, right, the strengths of these hidden interactions when we build genetic circuits. So that's the analysis and modeling part. But a very interesting question comes about when you ask, how can we make genetic modules robust to hidden interactions? So on the left-hand side, you have the system without hidden interaction. On the right-hand side, you have the system with hidden interactions. So the question that we are asking is that if you look at any one of these nodes, is a genetic module with inputs and outputs. And now what's happening due to these hidden interactions is that it's taking WI's perturbations that are deriving from the rest of the network, right, demanding resources. And also this genetic modules is applying a disturbance to the rest of the network. So the question we are asking is, can we design a biomolecular feedback controller that performs disturbance attenuation, meaning it makes the response of the genetic module to desired regulatory input independent of these perturbations? So since these WIs are not arbitrary signals, but they come from the network, the problem is really twofold. On the one side, for the isolated system, we want to see how we can design biomolecular controllers to make the response of the module robust to these fixed WIs. The second one is, can we still perform well regarding disturbance attenuation if these WIs, right, are part of this network of hidden interactions? So starting with one, this seems a rather trivial problem stated as this, but the reality is that it's not that trivial. So if one were to build this controller in a traditional engineering system, the first thought would be, well, let's try to put integral control in there and see if we can get perfect disturbance rejection. The problem with this is that in biomolecular systems, it is very difficult to engineer integrators because of unavoidable dilution in vivo. So cells grow and divide, and as they divide, all the molecules dilute, and therefore there is always to be a leakage to any integrator that you can come up with. So then the question is, we cannot send growth to 0, so we can never really put gamma to 0 here. The only option that we have as an attempt is to increase the speed of all controller reactions. So you get a situation where you have an integral controller with a quasi, so it's a quasi-integral controller because you have leakage, but the integral part is very fast. Of course, stability is not always guaranteed for these systems. So, but you can prove that if for gamma equals 0, so in the ideal integral control case, if the closed loop system were locally exponentially stable, then your output y becomes independent of w as epsilon is decreased. Okay. So then the bottom line for this is that we needed a biomolecular implementation that could be realizable by fast controller reactions, compare again to your regulatory input, and that could compute the difference between two signals, right, V and Y, and integrate it. So there was a lot of effort in the community in the past 5 years in order to try to figure out what biomolecular implementations could implement this integral function. And there is also a very recent beautiful paper by Mustafa Kamash group that appeared in Nature last year that is proposing one of these implementations. So in our case, the way we implemented this for our problem is called through sRNA silencing. So this is your genetic module, and you're trying to make your output protein respond to the regulatory input despite ribosome availability is changing, okay, for your translation rate. So what we did, we engineered a small RNA that could come back and eat essentially the mRNA in question and get destroyed together. So if you look at the simple ODE model, you can check that the speed of these reactions comes from free, because these sRNA interactions are much faster than protein production and decay. And we can engineer fast productions for these species M and S, essentially by engineering the promoters. So once you write the system this way and you make a change of coordinates, you can see that the quasi-integral control structure appears. You can prove local exponential stability when gamma is 0, and therefore you know that the system will work as almost an integrator. So here I'm showing just an example when you apply a disturbance. It's just simulation that show that after disturbance application, with epsilon really small, the system goes back essentially to the pre-stimulus condition. So this was a good starting step. I have to mention that the time-varying version of this problem, it's only partially solved. So if you're interested in learning more about this, let me know. But here it's an interesting question. With time-varying inputs, meaning disturbance and reference inputs V, can we still attenuate the effect of disturbance as the time scale separation epsilon, right, between the inputs and the controller goes to 0? So this is not obvious, okay? But a tempting observation is that if you look at the system, okay, then when epsilon goes to 0, you would be tempted to say that essentially the only solution becomes y equal to v, which is independent of w. So if you look, however, at the boundary layer dynamics, which is essentially the fast, what you think it's the fast dynamics of the system, has a Jacobian that is singular everywhere. So there is only one fast mode in the system, despite the fact that the system looks almost like in standard singular perturbation form. So what it turns out is that this system is called a singularly perturbed system, and there is a rich literature about these interesting structures. So essentially what you have is that if you have a system in supposedly standard singular perturbation form, and you take it into the fast variables, and you look at the Jacobian when epsilon goes to 0, essentially in standard singular perturbation, you would expect that J has exactly Q0 eigenvalue, which is the dimension of the slow subsystem. In an SSP problem, J has more than Q0 eigenvalues. Now, the good news is that if the zero eigenvalue has algebraic and geometric multiplicity that are equal, one can find a change of coordinates to take the system in standard singular perturbation form where the Tikhonov theorems can be readily applied. But fortunately, this is not the case in our case. We had the geometric multiplicity 1 while the algebraic multiplicity was 2. So how can we deal with this problem? So here's just to give you an idea that sometimes when you build these systems and then you analyze formally their dynamics, their structure, it's not quite trivial to address by looking at existing tools. So essentially the idea is that if you assume that you have a system of this form, right? So we are looking at the linearization only, and that's how much we could go for at this point. So if the zero eigenvalue has an algebraic multiplicity equal Q plus 1, the geometric multiplicity is Q, so there is just a one-dimensional discrepancy between the two, and then all other eigenvalues have negative real part, then you can prove that you can transform the system into a reduced system with just Q plus 1 dimensions, but the reduced system is epsilon dependent. And you can see here that the one-dimensional added system here, it's almost an integrator, okay? So what you can prove is that if you have suitable controllability assumptions between the two subsystems here, then you can prove that essentially this reduced system is square root epsilon close to the original system. And one way to prove it is, for example, to use the ISSS small gain theorem. And you can check the paper if you're more interested in the details. But nevertheless, now that we have this epsilon dependent reduced system, what you can do, you can go back and reason on it and see if we can make the output Y independent of W, okay, which is our original problem, the disturbance, which is now time varying, and the regulatory input, which is also time varying. So we have essentially a theorem that says that under additional technical assumption where all the input derivatives are bounded, essentially you are allowed here to set epsilon to 0 and say that essentially this is square root of epsilon. So Y is going to track V and is going to be independent of W, okay? And here is just a simulation going back to our simple model before that shows that once you apply a time varying W, as epsilon becomes smaller, essentially you're able to attenuate its effect arbitrarily. So now we go back to the experiments, okay? So now that we had a good grip that if we can make this system, right, with this sRNA interaction with fast enough reactions, then we went back to our original problem. So we have this genetic module, we have the disturbance W of t, which is your depletion of resources, if you will, which is imparted by another module, which is taking away resources or fighting for them. So this is the experiment, or one of the many experiments that shows that when you are activating this resource competitor and therefore it's applying a load to these cellular resources, here you have that the output of your genetic model of interest goes down, okay? While of course the output of your resource competitor goes up. So with that, then we asked, so now what if we put our feedback controller in here? So at this point, we had the regulated module, okay? That is essentially what I described before. We still had the resource competitor being activated at the same time, but now you see that the output of your genetic module is suffering at the very beginning, but then it's adapting through a rather expected adaptation response. And here just showing the steady state errors between the Y and the pre-stimulus value. And you can see that as we increase the disturbance, the closed loop system, which is this system, it's sustaining this perturbation much better than the open loop system that is being affected quite a lot. So this is just to show also the distribution. So this talk is focused on deterministic behavior, but of course we did analysis also on the stochastic behavior, and you can prove that your feedback controllers is not adding in any noise to the system, which is actually a concern when you have noisy molecular reactions. So as a last step, right, we go back to our original problem, which was network disturbance decoupling. Okay, so at this point, we have designed within each one of our nodes, right, a biomolecular controller that it's making sure that if I have a reference, right, and an output, right, the reference to output response is robust to perturbation Wi as this parameter epsilon is decreased. Now the big question is, can we still guarantee that if now my Wi is coming back from the network, right? So what happens as I decrease epsilon? In principle, right, what may happen is that as I decrease epsilon, I may increase the disturbance extent, right, to the network, and then I may increase Wi. So we need to establish essentially if there is an epsilon independent bound, right, in this loop here that we can place for Wi. So focusing on this, then what is that you can say? So here is just an example of the things that go wrong. So we had an example with 3 identical genetic modules in our resource conservation. And what this example shows is that each one of these genetic modules has the feedback controller that I showed you before, where you can decrease epsilon. If the reference inputs are small enough, you can see that as epsilon is decreased, you can attenuate the steady state error due to W, okay, arbitrarily. But if the reference inputs start to be too large, as you decrease epsilon, you lose the ability of attenuate the perturbation, okay? So why is this happening and how we can tamper with that? So here is the way we solve this problem. So we have this system, sigma I, which depend on this small parameter epsilon that have this disturbance attenuation property. And if you assume that this epsilon, sigma I epsilon can be reduced to input to state monotone systems, for example, via singular perturbation, which is often true because our controller, as we said, is fast, so it can be often reduced to its slow manifold. And the interconnection map is monotone, then you can use the rich theory that is available for monotone dynamical systems in order to prove that essentially if the discrete iteration that happens here can be bounded in an epsilon independent box, okay, then you have the network disturbance decoupling property. And we can prove that this property is true, meaning this iteration converges in an epsilon independent box only when the reference input Ri's to your plant are bounded in a set, which we can compute computationally, and it depends on the structure of the system and on essentially the parameters of the system, many of which we can tune. So in summary, here it's what I told you about today. Here's the picture. So we have been looking at problems of hidden interactions due to cellular resource competition. We have been looking at direct loading problems in between modules as example sources of how modularity fails when you engineer these systems. And we have proposing an engineering framework for essentially isolating these genetic modules from these perturbations. I show you two stories. One is what we call the load driver, which is this insulation device that performs disturbance attenuation based on timescale separation. And then there is the resource decoupler, which is designed through this quasi-integral control architecture, where essentially you have a fast time scale of a system that has an integrator inside in order to defeat the dilution or integrator leakiness. So that said, where do you go from here? So I have been focusing on these two problems, right? Intermodule loading and loading to cellular resources. There are many other problems. So for example, I haven't talked at all about how these modules often interact with the cellular genes, right? With the circuits that creates a whole added layer of perturbations that can affect your module. And then there are many more issues that people are studying and are developing control theoretic solution to tackle them. So here I just put a few. So laboratory conditions. So many of the circuits are really fragile to things such as nutrients, temperature, pH of the media, the cell type strain, right? If you change type of bacteria or type of either specific bacterial strain, things will change, the growth phase, and there are mutations. So many times you design a circuit, sometimes when you operate it for long, 1 or 2 days, you find out that the genetic sequence is different from the one you designed. So it's as if you are operating a computer and all of a sudden the computer transforms in a TV, right? So there are these sources of mutations. There is intense research to figure out on robust design approaches to prevent them taking over. So I just want to thank the people that contributed to this research. So here on the left, you have students that left my group or postdocs that left my group that heavily contributed to this research. There is Andres Georgi at NYU Abu Dhabi, Hati Chang at Harvard, Jose Jimenez in UK, and John Yazbek, and then my long-term collaborators at MIT and NEU. And then I will stop here, and if there is time, I'm happy to take any questions.
All right, so thanks, Domitilla, for your time and for your great talk. So Merve needs to leave in a few minutes, so I would like to lead questions. So to ask questions, basically, you can unmute yourself, or if you don't want your voice to be recorded, since we are going to post it to the YouTube, you can directly write your question to the chat. So Domitilla, so sometimes our audience is shy, and they, based on my experience, instead sometimes they don't, they mainly ask questions through email. Mm-hmm. So can we show your email if
Absolutely.
possible?
Yes. So I can put it in the chat.
That would be awesome.
Yeah.
So Domitilla, thanks again one more time. So and for participants, we also thank them, and if they have any questions, then they can directly email it to you.
Okay, sounds good. Thank you so much, and goodbye, everyone.
Thanks, Domitilla.
Thanks. Goodbye, everyone.